we talked about the light reactions. And the light reactions basically is going to provide us the energy to create ATP and NADPH. When we have ATP and NADPH, we are going to actually use them in a cycle called the Kelvin cycle. The Kelvin cycle is the place where the plant actually called an autotroph, because autotroph is an organism can create its own food, and plant can create their own sugar. In our case, they are going to create glucose molecules. And in order to create glucose molecules, we need to have a process of something called carbon fixation. Carbon fixation is where we take carbon dioxide from the surrounding, from the air, in the form of gas, and we are going to fix it in a solid structure. In our case, it's going to be the last product of the Kelvin cycle, which is each time is going to be recycled and fixated to carbon dioxide. So we are going to add carbon dioxide because, as you remember, sugars, glucose, have a carbon backbone, and we have to have carbon inside. We have to incorporate the carbon. In order to do so, we need to have energy. And we get the energy from the ATP and the NADPH we got from the light reactions, from the sun. So let's try to see what are the dark reactions, the Kelvin cycle, in a very some sort of introduction. You are going to see that we are going to have 6 carbon dioxide, because glucose have 6 carbons, and we are going to need 12 ATP and 12 NADPHs in order to create 12 molecules of something called pGAL. pGAL is the main molecule when it comes to this cycle. So through the fixation of 6 carbon dioxides with 12 ATP and 12 NADPH in this cycle, we are going to get 12 molecules of pGAL. Now these 12 molecules are going to divide it into two groups. We're going to have 10 molecules of the pGAL, which are going to continue the cycle. And these 10 molecules are going to become something called RAB-BP, or rubisco bisphosphate. Rubisco bisphosphate is the last product of the Kelvin cycle, and it's the product that is going to get the carbon dioxide once again. Similar to the Krebs cycle, when we have the last molecule of the Krebs cycle combined with acetyl-CoA to continue the cycle, same here, we have rubisco bisphosphate combined with the carbon dioxide to continue the cycle. So we are going to have 10 of these molecules continuing the cycle. However, we are going to have 2 of these 12 molecules, because we said we have 10 pGAL, and we produce 12, which means we have 2 left. We are going to take 2 molecules of pGAL, and we are going to combine them to create glucose. How can we combine them to create glucose? It's because pGAL is a 3-carbon molecule with a phosphate group on it. And as you remember, glucose is a 6-carbon molecule. The fact that we have phosphate group on this 3-carbon molecule will allow us to get enough energy to combine the molecules, 2 molecules of this together through a specific enzyme in order to create glucose molecule. So this part of the cycle is actually the part that is going to allow us to create the glucose molecules. And this part of the cycle is actually the part who is going to continue the cycle and to allow rubisco bisphosphate to continue to the next stage. Now I want to zoom into the cycle and allow you guys to actually understand what's going on in details. The Kelvin cycle actually occurs twice. Each Kelvin cycle is going to produce 1 molecule of G3P, of the sugar or pGAL. We have G3P, we call it pGAL, it's different names. We are going to have the pGAL. What are we going to do with the 1 molecule? We're going to wait for another cycle, because only when we have 2 pGALs we are going to be able to combine them to form sugar. So 1 Kelvin cycle is basically going to take half of everything I just talked about. So 3 carbon dioxide, 6 ATP and 6 NADPH, which is really important to remember. So in total, in order to create 1 glucose molecule, we are going to need double what you see here. So let's try to analyze, as a summary for this video, let's try to analyze the following metabolic pathway. So we are going to have carbon dioxide, which is going to be combined with the last product of the Kelvin cycle. Through a very important and high yield enzyme to remember, they are going to combine together into using Rubisco. By the way, Rubisco is the most abundant enzyme in the world. And you're going to get this very non-stable 6-carbon molecule. This very non-stable 6-carbon molecule is immediately going to break down into this molecule, which is called 3-phosphoglycerate. Now, through several steps, we are going to have the ATP phosphorylation and the NADPH, and we are going to get the intermediate all the way until we get to the PGAL, to the G3P. This part is where you have the diversion. One molecule, because it's one cycle, is going to become and wait for the next one in order to become a glucose. And the next one, in our case, the 5 other molecules are going to allow us to create Rubisco bisphosphate. And this molecule, again, going to combine through carbon fixation, through Rubisco, with the next carbon dioxide in the list. So one Kelvin cycle is basically what you see over here. And remember, we need to have 2 Kelvin cycles in order to have one glucose molecule, because we need 2 outputs, 2 G3P. A very high yield point to remember. Another very high yield point to remember is that you have 3 carbon dioxide entering at one time. So make sure you remember the following high yield points and make sure you understand the Kelvin cycle as well. Now when you have the ADP and the NADP+, they are going to go to the light reaction in order to gain the energy back, to be recharged again, in order to fuel and drive the Kelvin cycle. Remember that even though it is called the dark cycle, it requires the light in an indirect way in order to keep working. So even though it is called the dark reactions, you don't have dark reactions during the night because you need NADPH and ATP from the light reactions to drive this process.